Okay, well, sadly, I will not be talking about changes in distribution on the west coast of Canada. But should I go back there? Or do you want me to put on the microphone?
Or you can take one of those.
Can I just put this on?
Yeah,
It's probably the easiest.
just make sure it's not on mute.
Hello? Nope.
Is the green light flashing?
No, it says it's on. Yeah. Take this up. All right, can everybody hear me? Hopefully they can hear me through the microphone. I don't hear the microphone here, but anyways.
Your voice, for it to be captured with your PowerPoint on the Panopto requires being.
Okay, so we just assume that it's working.
There's a switch there. Yeah, that's on mute.
Ah, perfect. Oh, there we go. Okay, great. Okay, so I'm really glad that Tara introduced the workshop in the way she did, talking about sort of universal responses to climate change, because these are tackling these questions in an empirical way is the focus of my research. I'm primarily a buckets and boops empirical ecologist, and what I'm interested in doing is understanding how responses to temperature at the individual level can help us predict population responses to temperature. And so we've been talking a lot about this question of can we predict patterns of abundance and distribution into the future, and I would argue that this is actually a central challenge in ecology. Understanding what drives the abundance, distribution, and diversity of life on Earth is a grand challenge that ecologists have been trying to address basically since the discipline began. And even though we've been answering this question from a whole variety of perspectives, I would argue we still lack a coherent and mechanistic understanding of what drives these patterns. And those of us who study biodiversity, some of us tend to focus on what makes species different from one another, but I would argue that a complementary approach is to study what makes species similar. And one feature shared by all of life on Earth is metabolism. Every living thing must somehow acquire resources from the environment, it must transform these resources within itself, and then allocate these resources to the various processes underlying survival, growth, and reproduction. And this has really important implications for ecology because ecology is fundamentally metabolic. Essentially all ecological interactions, any one that you can imagine, involves exchanges of energy and materials. So everything from competition, predation, and facilitation. And one of the most widely observed patterns among ectotherms is that metabolic rates increase with temperature. And so plotted here is organismal metabolic rate on the y-axis, and on the horizontal axis is temperature, and it increases from left to right, even though it's plotted on this really wonky Arrhenius plot. What we see here from a survey of a whole bunch of different species of fish is that metabolic rates increase with temperature, and we can understand this persistent positive slope as these sort of the effect of unavoidable or unescapable physical constraints imposed by temperature. That is that the enzymes that catalyze the reactions underlying metabolism, they speed up with temperature. We might expect, though, that if evolutionary adaptation were somehow able to overcome the physical effects of temperature, this relationship would look like a flat line. But we see instead this positive slope, meaning that there are these persistent effects of temperature on metabolism. And this positive response of temperature on metabolism is one of those widely observed patterns among ectotherms. Not only do we see the signature of this positive slope across a whole variety of taxonomic diversity, but this slope, the value of the slope, consistently reflects the effect of temperature on cellular respiration, suggesting that these constraints scale from cells all the way up to ecosystems. And this is what we call metabolic scaling, when constraints operating at very fine levels of biological organization, like within cells, emerge in higher order processes up to ecosystems. And formalizing these relationships between temperature and metabolic rate and body size sort of forms the basis of the metabolic theory of ecology. And metabolic theory actually arose from macroecology, right? It arose from observing patterns at very broad continental scales, and from these patterns sort of inferring mechanisms that might be operating within individuals. And so for this reason, metabolic theory of ecology is a primarily empirical theory, right? It arose from observing patterns and then inferring mechanisms. And for this reason, it's sort of been criticized for maybe ignoring a bunch of potentially important dynamics operating within and among individuals, but nonetheless, it's proven remarkably successful at predicting large-scale patterns in nature. And so the focus of my work is to apply this theory to help us understand processes operating within populations. And so the metabolic theory of ecology explains how macroecological patterns in individual metabolic rates constrain processes at all these various levels of biological organization. And in theory, these constraints should operate at the level of populations. And in fact, for most of us, we're interested in predicting population-level responses to temperature. But I would argue that the testing and the development of this theory at the level of populations has been sort of the least well-developed, and really rigorous empirical tests have been lacking. And so today, the kind of driving question for my presentation and for my work broadly is answering the question of, does the temperature dependence of organismal metabolism that we observe across species, like the one that I showed you in that first plot, does this also drive dynamics within species? And to some extent, if there's a direct relationship between temperature and the need to make more ATP, then this could, in theory, apply equally as well to organisms adapted over evolutionary time, these patterns that we see in macroecological syntheses, but also to individuals within populations. On the other hand, it's really not at all clear yet that the temperature dependence of individual performance should translate directly to population dynamics, because we know that population dynamics are a function of feedbacks both within and among individuals. So this is where competition comes into play, density dependence, and species interactions. And so in my work, I focus on testing links between individuals to populations. And my approach is to take macroecological patterns as hypotheses and to use metabolic scaling theory to test some of these hypotheses, which I test primarily in aquatic mesocosms in the lab. And then ultimately the goal is to piece together these various lines of evidence to make predictions about abundance and distribution now and into the future. And so today I'm going to address three main questions that all center on this challenge of scaling from individual to population responses to temperature. The first question is, does temperature-dependent individual metabolism, does it scale up to population abundance? The second question that I'll address is temperature-dependent body size predictable based on metabolic demand? And then finally, I'm going to sort of break down one of the primary assumptions of the metabolic theory approach, which is that temperature remains constant over time, because of course we know it varies. And I'll ask, can thermal variation break down the relationship between individual and population level rates? Okay, so does temperature-dependent individual metabolism scale up to population dynamics? And here the question really is, if we understand how individuals respond to temperature, can this help us make quantitative predictions about how populations respond to temperature? And one way to do this is to essentially trace demography back to mass and energy budgets of individuals. And so what is shown here is the logistic model. This is kind of a core model in ecology. What we want to know is how do R, the intrinsic rate of increase, or population growth rate, and K, the carrying capacity, how do these vary with mass and temperature? And the effect of temperature on little r, the intrinsic rate of increase, has been widely studied. We generally know that hotter is better, that is that little r increases with increasing temperature. But carrying capacity has been pretty much entirely overlooked in terms of the effect of temperature on carrying capacity. And carrying capacity is actually a critical link between population dynamics and ecosystem and biodiversity patterns. And most attempts to scale from individual up to ecosystem responses to temperature have had to assume a certain dependence of population carrying capacity on temperature. Some studies, when looking at the food web level, have assumed a positive relationship between temperature and population carrying capacity. Others have assumed a negative relationship between temperature and population carrying capacity. And what we see from these theoretical studies is that this assumed relationship has really important implications for the effect of temperature on food web stability. But until now, these relationships have not been tested empirically. And so to answer this question, I'm drawing on theoretical predictions, part of metabolic scaling theory, that link the mass and energy budgets of individuals up to populations. And here's a prediction based on this theory. Oops, sorry. I don't know what I just did there. Which one's the laser? This one, okay. Here the prediction is that carrying capacity and the temperature dependence of carrying capacity should reflect the temperature dependence of individual metabolism like this. It should scale as a function of mass and temperature. And in particular, carrying capacity should decrease with increasing mass, and it should decrease with increasing temperature. And this might seem kind of counterintuitive. Why should population carrying capacity decrease with temperature? And the intuition here is that if resources are held constant across a gradient of temperature, metabolic rates will increase, metabolic demands will increase, and therefore, given a constant supply of resources, the total number of individuals that are able to be supported by a given resource supply should go down because each individual is fluxing energy and materials at a higher rate. So a system should be able to support fewer individuals. And in particular, this theory makes the prediction that as individual metabolic rate goes up with a certain slope reflected by this value EA, the population abundance at carrying capacity should directly reflect the temperature dependence of individual metabolism. So this is the prediction that metabolic theory makes. This has not been tested empirically. So I set out to test these theoretical predictions using a species of green algae that was originally isolated off the west coast of Vancouver Island. It's a coastal marine species called Tetraselmis tetraheli. You can see it swimming around here. It's highly mobile, meaning that it allows me to meet the assumptions of the logistic growth model, which are that everybody has equal access to resources. And all I did was I grew these temperatures over a gradient of temperature and followed them until they reached their steady state abundances. Okay, so the first, however, though, to test this theory, the first thing we know is how do individual metabolic rates depend on temperature. And so the prediction is that individual metabolic rates should increase, and in phytoplankton, the most relevant metabolic rate here is the rate of photosynthesis, or net photosynthesis. And consistent with theory, we see that individual metabolic rate, or individual photosynthetic rate, increases with temperature. Okay, so that's the first step here. The second step then is to use that temperature dependence of individual metabolic rate to make a prediction about how carrying capacity should decrease. And here this slope directly reflects my empirically observed temperature dependence of photosynthesis. Okay, so here's our prediction then, that carrying capacity should decrease with temperature. And here's what we find. So carrying capacity does decrease with temperature. The observations are plotted here in the dots. But here was our prediction, and our observations fall the slope here is much less steep than theory predicts, meaning that as temperature increases, populations are able to support higher abundances than we would have predicted based on the temperature dependence of metabolic rate alone. Okay, so our attempt to scale up from individual metabolic responses to temperature to a population response did not work. That is, we were not able to quantitatively predict carrying capacity as a function of temperature. So either the model is wrong here, or there's something going on that we didn't account for. And what that thing here is that body size also decreases with temperature, concurrent with the decline in abundance. And so what I've plotted here is cell size at equilibrium as a function of temperature. And we see that body size, or cell size in this case, decreases by about 2% per degree increase in temperature. And this is consistent with a very widely observed pattern among ectotherms known as the temperature size rule. So if we go back to our original metabolic scaling theory predictions, what we see is that they include mass and temperature, but don't allow mass itself to change with temperature. So don't account for the interaction mass itself changes with temperature. So what I did was I basically expanded the theory to allow mass itself to depend on temperature. And here this parameter beta just that reflects the empirically observed decline in body size with temperature. And what we see is that if we allow the temperature size rule, if we allow mass to change with temperature, we can actually predict quantitatively the decline in abundance. So what I'm showing here, this prediction that includes the temperature size rule, allows us to make quantitative predictions of abundance at carrying capacity, demonstrating that the temperature dependence of abundance directly reflects the temperature dependence of photosynthesis and body size. Okay, so I started off by asking, does temperature-dependent metabolism drive the dynamics of populations? And I argued that we didn't have any empirical evidence yet that this link could be made and that these predictions could be quantitative. I showed you now that yes, individual responses to temperature scale directly to populations. And this is the first empirical support for this often assumed negative relationship between carrying capacity and temperature. But importantly, we see that declines in body size effectively reduce per capita resource demand, right? Reduce the individual demand for resources, and therefore reduce declines in abundance. Okay, so now that we see that changes in body size are clearly an important part of this task of scaling from individual to populations, the next question I want to address is, can we actually predict that decline in body size? So we saw that it happened, but now I want to ask, is that decline actually predictable? So the temperature size rule, like Tara mentioned earlier this morning, is known as sort of a third universal response to warming alongside shifts in distribution and changes in phenology. And while it's so widely observed, I would argue we still don't know what explains this pattern and whether there is maybe a general mechanism that underlies this pattern across a wide range of taxonomic diversity. And as Tara mentioned, and as many of us are familiar with, there are certainly many competing hypotheses for what explains the temperature size rule. And there are certain, there are a variety of models that have been invoked to predict it, but I would argue there's really only one that's general across basically all of taxonomic diversity. And what this model suggests or predicts is that reduced body size can be explained by increasing metabolic demands. And so the model that I'm testing here in this context is what's known as the supply-demand model, and it's developed by John DeLong. And the predictions that this model makes sort of rest on the premise that individuals must somehow uptake resources and allocate them to these various processes. And basically, the prediction that this model makes is that individuals start off at some body size M sub B, and they grow throughout their lifetime up to some asymptotic body size, M infinity. And basically, individuals will mature, or they will be adults at a certain point in which their demand for resources is directly balanced by the supply of resources in the environment. So it's a very simplistic model that basically says individuals will grow up to a size at which they can basically become as big as they can given the available resources, but not too big such that they run out of resources. And so this model has been tested in the context of protists, but has not yet been tested in the context of the temperature size rule. And so one prediction that we can derive from this model is that if we think about two temperatures here, this demand curve in blue being a cool temperature, and this curve, or this line representing a warm temperature, what you can see here is that if temperature causes metabolic demand to go up, then the point where these two curves meet, here it will be a smaller body size. So basically what this model predicts is that increases in metabolic demand, if resources are held constant, should lead to a smaller body size. So my approach was to test this model empirically using Daphnia, which is a species of freshwater zooplankton, a model organism for understanding life history. And my approach was to grow up these individuals in single beakers in the lab. And so they're essentially given like penthouse conditions of unlimited resource supply, but resource supply is held constant through time. And I grew them at a range of 5 temperatures, and I measured their body sizes at each one of their first till their third clutches, and estimated their asymptotic body size. I also tracked their fitness, and I'll tell you why in a minute, but basically the general motivation here is asking the question, is the temperature size rule like a very, very widely observed case of phenotypic plasticity that everybody seems to do, all ectotherms, or are there also fitness benefits to being smaller when it's warmer? That is it under selection, and does it come with a fitness benefit being smaller when it's warmer? So the first prediction that this model makes is that metabolic demand, or this is a sort of general metabolic theory prediction, that metabolic demand should increase with temperature. And I tested that empirically, and so what we see here is that when you put individual Daphnia into tiny, tiny little respirometers over a temperature gradient, their individual metabolic rate goes up as the temperature increases. So we have empirical evidence that, not surprisingly, metabolic demand increases with temperature. If resources are held constant, then this leads us to the prediction that maximum body size should decrease with temperature. But importantly, we have a quantitative prediction for this. We should directly be able to predict this based on this slope of the temperature dependence, and also this b, which is an allometric scaling exponent, which basically describes how metabolic rate increases with body size. And this also I determined empirically. So we have a prediction then that maximum body size should decrease with temperature. And here's what we find. Consistent with our prediction, maximum body size decreases with temperature, and the slope of this relationship is predictable based on the temperature dependence of individual metabolic rate. So you can see that, I mean, it's because this black line falls within the confidence intervals, the dashed lines of our prediction. So here we have empirical evidence that the temperature size rule and the response, the decrease in temperature, is quantitatively predicted based on the temperature dependence of individual metabolic demand. So while those two observations are consistent with the supply-demand model, they're not a critical test of the supply-demand model. In order to make a critical test of the supply-demand model, we need to find evidence for a trade-off between maximum body size and growth rate, or maximum body size and metabolic rate, in this way. That is, that maximum body size and individual growth rate should trade off with a slope of -1/b, and we can get into that later. But if we find support for this, this would be empirical evidence that individuals are basically have some sort of physiological compensating mechanism that allows them to, as I mentioned, grow up to a certain size given resource availability. Okay, so here's our prediction on the left, and here's what we find. Plotted on the y is maximum body size. This is your L infinity that we've been talking about so far. Individual growth rate, this is your K, estimated from a von Bertalanffy model. And here we see that maximum body size, there is a trade-off between maximum body size and individual growth rate, and that trade-off is quantitatively predicted. So we now have empirical evidence that not only does body size decrease predictably, but this decrease is attributable to this trade-off between maximum body size and growth rate, meaning that metabolic demand drives decreased body size at higher temperature. Okay, so I now want to ask more broadly, what are the fitness consequences of being smaller when it's warmer? And this question has actually formed the basis of a life history puzzle that people have been asking for decades. And the puzzle is, we know that being bigger is generally better, right? Larger body sizes are generally associated with higher fecundity. So smaller body sizes, therefore, associated with the temperature size rule, should be associated with lower fecundity, which we know is probably bad for fitness. On the other hand, we know that hotter is better. That is, that faster metabolic rates are associated with shorter generation times, and a shorter generation time will lead to higher fitness. And so there's sort of this potential conflict between the direct effects of temperature on biological rates and the indirect effects of temperature on body size. And resolving this question is critical to understanding the fitness implications of the temperature size rule. So so what I'm plotting here is the intrinsic rate of increase, or little r, as a function of body size, and each different color here represents a different temperature. And what we see here is that we find evidence for hotter is better. This is consistent with what is widely observed. So as we go from purple to yellow, this is 10 degrees to 27 degrees. Growing at warmer temperatures leads to higher intrinsic rates of increase, but also smaller is better in this case. So there's a persistent negative slope here, meaning that within a temperature, variation independent of temperature is associated with higher intrinsic rates of increase. So what this suggests then, just to summarize this portion of the talk, is that first of all, we found empirical evidence for the temperature size rule being explained by increased metabolic demands associated with warming. But the fitness consequences are that being fitness consequences of being smaller when it's warmer is that the positive effects of warming on generation time outweigh the negative effects on fecundity, meaning that being smaller when it's warmer leads to a fitness benefit. Therefore, suggesting that the temperature size rule might not only be a phenotypic, a case of phenotypic plasticity, but that's also under selection. Okay, so the last question that I want to ask now is basically breaking down this assumption that temperatures remain constant over time. And in both of the studies that I've shown you so far, and in pretty much the vast majority of studies that investigate temperature effects in the lab, temperatures are held constant, right? We program incubators, we program them to maintain a certain temperature. And this is the conditions under which this theory was developed. But obviously we know that in nature, temperatures are not constant over time, right? Organisms live in variable environments. And temperature and other environmental conditions fluctuate from day to day, from season to season and across the globe. And the way in which we characterize organismal responses to temperature is via thermal performance curves like the one shown here. And so what we see here is that, and what we know is that organismal responses to temperature are almost always nonlinear. And so for those of you who aren't familiar with the thermal performance curve, what this shows is that performance could be growth rate, usually increase from some minimum temperature up to an optimum temperature for performance, and then decrease down to a maximum temperature. And we often use these estimates of thermal performance and the thermal niche to make predictions of population persistence and range shifts and abundance under scenarios of climate change. But because these curves are nonlinear, and because temperatures change over time, any prediction of population persistence or range shifts under climate change must explicitly consider variation in temperature over time. And this is a basic fact of nonlinear averaging, known as Jensen's inequality, which shows us that if temperature varies over time, if we're in a portion of the thermal performance curve that's increasing, that is, that has a positive second